And so with that, we're moving to our next speaker, Peter Rupprecht. Peter is, like many people in the field of computational neuroscience, is a physicist by training. And then he did his PhD in experimental neuroscience and is now a postdoc in the lab of Friedrich Helmchen in Zurich. And so he'll be talking about deep learning toolbox for noise-optimized generalized spike inference from calcium imaging data. Peter, you're muted.
Oh, better now?
Much better.
Okay. So I think we all agree that calcium imaging is a really powerful method, also because it's visually stunning. However, I think the readout of neural activity is quite indirect, as Josh already mentioned before in his talk, and therefore hard to interpret. So for example, what do these transients really mean? So what does it mean in terms of spiking activity? So many algorithms have been developed, which basically infer the firing rate based on these slow and indirect signals like sweet2p from Kaiman, from MLSpike, as mentioned by Josh, from peeling, Emcee, and so on. It's a huge range of algorithms. However, when I first entered the field, I was really a bit depressed by these algorithms, because in the beginning, when I tried to apply those algorithms on my own data, I was not so confident that I got really usable results. First of all, I didn't know how to tune the parameters of the model. Second, the results seem to be often very arbitrarily scaled. Sometimes I got results like 100 hertz firing rates when it was obviously not the case, just a bad fitting. And the key question I was asking myself was, how do these algorithms work on my own data? Because these have been designed on one or two data sets which have been recorded by somebody else. But how do these algorithms translate and transfer their power to my data? And obviously, so can I believe these results? And the way to address this question is to look how these algorithms generalize to new unseen data sets. And this was basically not possible until recently, because there were not enough data sets available. So that's basically a key result of my approach, to basically assemble a lot of data sets. Some of them had been acquired already before by the Genie project, by the SpikeFNet project, and also by the Allen Institute. Others I have re-extracted from existing publications, and yet again, others I have recorded myself or in collaboration with other people. Together, this makes up a database of 27 single data sets with different conditions, totaling more than 300 neurons and many brain regions, many different calcium indicators, spanning a huge variety of conditions. Of course, now I could have used one of the previously mentioned model-based methods, basically fit the parameters of these models. However, I think that's really not the way to go if you have a huge data set with millions of data points. Therefore, I used a deep network and basically used a local environment of the calcium trace to predict the spiking probability or the spike rate for this time point using this deep network. This is actually a very simple and small network, so it's so small that I can run it on my laptop without any issues, which is really rare for real-world applications and very instructive if you want to go into deep learning without huge GPUs. So I want to highlight only two aspects of this algorithm. First is how it generalizes to different data sets. So basically, each row is a model trained on one data set. And the columns indicate how well does this model for data set 2 predict the spiking activity for the data sets which are different from this data set. And as you can clearly see, there's not a lot of structure. And this is surprising because we organized these data sets according to calcium indicators and species. And the fact that we don't see a lot of structure in this predictive matrix indicates that it is not possible to predict from the calcium indicator or from the indicator species, from the species, how well the algorithm will perform on unseen data. So to circumvent this limitation, which is basically simply a fact which we found, is that we basically trained a single deep network on all of the data sets. And we found that this network, which is basically using all information available in all data sets, performs much better than any of the single models. The second, this is an interesting observation. The second aspect I want to highlight is that for population calcium imaging, the delta F over F calcium signals are typically quite noisy. And whereas for the ground truth that is underlying all these data sets is really assumed in single neuron, which is recorded at very high signal to noise ratio and temporal resolution. So, however, if you train the model on these very high signal to noise ratio data, you basically get only crap out of it because the network never has seen these noisy signals and therefore assumes, oh, okay, all of those are signals. However, if you add some noise on top of the ground truth and train the network again on this ground truth, then the predictions are much better. So it's basically very important to noise match basically the ground truth to your target recordings. And for the rest, I will just summarize some key aspects of our algorithm. So first of all, the network is able to retrain itself based on noise levels, which it detects automatically. There are no parameters which have to be tuned because it's a universal model which works for all of the investigated data sets, with some caveats, of course. We found that the performance much better than any of the model-based algorithms which we tune to all of our data sets as well. In addition, the result is properly scaled in terms of spike rates. And of course, the most important aspect is that we have tested the generalization to unseen data. And we have not, which allows us to estimate the performance on unseen data, which are basically data that you and I have recorded by ourselves. And then we can predict how well it will perform on those unseen data sets. Yet another reason why you should give it a try to use CASCADE, which is the name of the algorithm, is basically that you don't have to tune parameters and you don't have to install it. So basically you can just click on the link in a GitHub repository and you will basically be able to open a Colab notebook and just let it run by clicking on the play buttons here and there. And so it's quite easy to use, I would say. With this, I'd like to thank the people involved, for instance, Rainer Friedrich and Friedrich Hempchen, who supervised the project, and then Edwin Hoffman, who contributed to the toolbox, and many other people from Tokyo, Yale, Berkeley, London, and the Allen Institute, who contributed these very rare and important ground truth recordings. Of course, I'd like to also mention some open data sets which I used for this project and my funding sources. Thank you.
Awesome. Thank you so much, Peter. Are there any questions? Please submit your questions. Do any of the other two panelists have a question?
Yeah, I have a question. So if you compare the spike rates that you read out using CASCADE from the Allen two-photon brain observatory, the rates are over an order of magnitude lower than what we get when we record electrophysiologically. And of course, some of this, like I said, is probably due to the sampling bias of EFIS towards higher firing rate cells, but there's still a pretty big discrepancy there. So how do you account for that?
I'm quite confident that the firing rates are quite realistic. That's what I would say. Just to give some context, so I applied this algorithm also on a data set which was used by Josh Siegel in his talk as well. And based on that, I inferred typical firing rates for all of those neurons. And typical firing rates were around 0.1 or 0.2 hertz. And this is based on calibration experiments, which were also recorded in the Allen Institute. So I think it's as good as it can get, because it's even using the same transgenic lines in some cases. So I'm pretty confident that it is quite precise. Or what do you think about this, Josh?
Well, I mean, we almost never record cells with firing rates of 0.1 hertz. It's almost always 0.1 hertz and above, up to 10, 20 hertz in our ePhys experiments. And so somehow either it's missing spikes, or the training was biased in some way. Like I'm not really sure how to account for such a large order of magnitude difference in firing rate.
Yes, I think that's an open question. But for the bias that you mentioned, I think we measured this bias based on our ground truth, and I think it was quite small. So there's not an order of magnitude bias, but maybe at most 50%.
Yeah.
Or I think one reason could be that there are many. When I looked at the LN data set, many neurons seem to be simply completely silent. There was nothing happening at all. I think maybe these are simply not recorded at all in the ePhys data.
Yeah, that's the sampling bias that we're trying to quantify.
Great. Are there other questions? I have a question. So did you look at or inside your network to figure out how the network manages to do the inference?
Yes, actually, since the network is so small, it was quite easy to do. Basically, and here is the look inside end of the network. So here's basically the input, the delta F of F trace. Here is basically the inferred firing rate. And here you can look at the intermediate layers at the activation. And that's what you see immediately. So it's basically, I think it's interesting because you can visualize the entire network just on the half slide of a PowerPoint. So it's relatively transparent. I think maybe you're thinking more about understanding the receptive fields. And this is something I haven't done systematically, but maybe it's interesting. Would be an interesting idea.
Yeah, I was thinking more. Yeah, you call it receptive field. I would probably call it more like a transfer function or something like that. But or like essentially the relationship that allows you to transition from your input to the spike rate, right? Yes, you should be able to analyze that and get like an approximate close form or some relationship that's approximate that you can get out of the network.
You mean from end to end or from single units?
Yeah, end to end.
I think of course it's not a linear function because we tried if we apply the linear network to the same application, it doesn't work at all, or not by far not as good. I think it's quite tough.
So, but don't you expect to get the generative function that Josh was talking about out of this?
This generative function, which he mentioned, this is from the ML Spike toolbox. It's quite, I would say, it's stereotypic, and it's basically can be optimized by using a mean approach. However, the calcium signals are per spike per neuron are quite variable across neurons. I think to account for this variability, I think it's not enough to consider the mean transfer function. I think it's missing a lot of important aspects of the network. But this could be done, of course, yeah.
Cool. Okay, thank you so much. I think we